Bismillah ar-Rahman ar-Rahim. Assalamu alaikum, dear students. Welcome to your course Environmental Microbiology, ENS-224. As you know that we had started the role of microbes in soil. We had studied the carbon cycle, and today we will talk about the microbial activities in soil with this perspective of nitrogen, or simply the nitrogen cycle. The discussion agenda for today class is the nitrogen cycle, in which we will study different steps in the nitrogen cycle, that is the nitrogen fixation, ammonification, nitrification, and denitrification. First of all, if we talk about the nitrogen gas is the most abundant component of the atmosphere, that accounts for nearly 79% of the total air volume. As we know that this extensive reservoir in the air is largely unavailable to most of the organisms. Only about 0.03% of the Earth's total nitrogen is combined, or simply we can say fixed one way or another way, in some other forms such as nitrate, NO3, nitrite, NO2, ammonium, NH4, and some other organic nitrogen compounds such as proteins and nucleic acids. The nitrogen cycle is relatively more complex than other cycles, as we had studied the carbon cycle, because it involves such a diversity of specialized microbes to maintain the flow of the cycle. Big higher plants can utilize the nitrate and ammonium, while the animal receives the nitrogen in the organic form from either plant or other animal. The nitrogen cycle basically includes four basic types of reaction. What are these reactions? These reactions, first of all, there is the nitrogen fixation, the ammonification process, the nitrification, and the denitrification process. We will study all these in detail, that how these steps or these reactions play a role in the cycling of nitrogen. Here in this figure, it is a simplified event in the nitrogen cycle. Here you can see that in nitrogen fixation, the gaseous nitrogen is acted by the nitrogen fixing bacteria, which give off ammonium. That ammonium is converted to nitrite and nitrate by the nitrifying bacteria in the nitrification process. Plants, algae, and bacteria use nitrates to synthesize nitrogenous organic compounds like proteins, amino acids, and nucleic acids. These organic nitrogenous compounds are used by animals and other consumers. After nitrification, if we talk about the ammonification, nitrogenous macromolecule from waste, or simply we can say the dead organic matter or the dead organisms, are converted back to ammonium by ammonifying bacteria. This ammonium can be either directly recycled into nitrates or returned into the atmosphere, or through a denitrification process. They are returned to the atmospheric molecular nitrogen or nitrogen gas formed by denitrifying bacteria. So this was the simplified event of the nitrogen cycle. Now further we will go towards in detail. Uh, the first one is the nitrogen fixation. As you know that the biosphere is most dependent upon the only process that can remove the gaseous nitrogen or the molecular nitrogen N2 from the air and convert it to a form that is usable by the living things. And this process is called nitrogen fixation. The nitrogen fixation is basically the beginning step in the synthesis of virtually all nitrogenous compounds. The nitrogen fixation is brought about by the nitrogen fixing bacteria that is present in the soil as well as also present in water through a small amount nitrogen fixation. Also a small amount is also formed through a non-living process such as lightning. Lightning also fixes some atmospheric nitrogen, but the amount is too much small. The nitrogen fixers that are bacteria have developed a unique enzyme system that are capable of breaking the triple bond of nitrogen molecule. As you know that nitrogen forms triple bond. So when one nitrogen combine with other nitrogen, so in between these two nitrogen is a triple bond. So the nitrogen fixers have a unique enzymatic system that is capable to break that triple bond. If it breaks down, it reduces the nitrogen atoms, and this process is an anaerobic process that requires an expenditure of a considerable amount of ATPs. So after breaking, the primary product of nitrogen fixation is the ammonium ion. The nitrogen fixing bacteria live freely or in symbiotic relationship with plant. In coming slide, we will talk about how it forms a symbiotic relationship with plants. Among the common free living nitrogen fixers are the aerobic Azotobacter species, Azospirillum, and certain members from anaerobic genera, that is also Clostridium species. Similarly, the other free living nitrogen fixers are also from the cyanobacteria, that is Anabina and Nostoc species. Yeah. How the nitrogen fixation occurs, as we have studied in the previous slide. Basically, nitrogen fixation occur in root nodules, or simply we can say natural fertilizer factories. As you know that a significant symbiotic association between rhizobia, that is bacteria in the genera Rhizobium, Bradyrhizobium, and Azorhizobium, that associate with legumes plants such as soybean, peas, alfalfa, and clover. Or simply we can say those plants that characteristically produce seeds in pods. Or further, we can, I told you, we can also say these plants as legumes. But how the association begins? Basically, the association begins with the infection of legume roots by these gram-negative rod-shaped bacteria, like as I told you, Rhizobium, Bradyrhizobium, and Azorhizobium, cause the formation of special nitrogen-fixing organs that are termed as root nodules. Yes, you can see in this figure the nitrogen fixation through symbiosis in sweet clover plant. First of all, as you can see in this image, the event leading to the formation of root nodules, cells of bacterium Rhizobium, as you can see here, attach to the legume root hairs and cause it to curl. This you can see here, it's curl of the root hair. Invasion of the legume roots proper by Rhizobium initiate. Here you can see in this figure, the formation of the infection thread that spread into numerous adjacent cells. After that, the presence of bacteria in cell cause nodule formation, and here you can see the mature nodule that have developed in the clover plants. Nodulation begins when the rhizobia colonize specific site in the root hairs, as you saw. From there, the bacteria invade deeper into roots and induce the cell to form a tumor-like masses. Basically, what the bacteria actually do, the bacterium enzyme system supplies a constant source of reduced nitrogen to the plant. And what actually plant do for the bacterium, and the plant furnish nutrients and energy for the activities of these bacterium. So the legumes uses the ammonium to aminate. Aminate means add an amino group to various carbohydrate intermediates and thereby synthesize amino acids and other nitrogenous compounds that are used in plants and animal synthesis. Basically, this plant bacteria association have a great practical importance, especially in agriculture, because an available source of nitrogen is often a limiting factor in the growth of crops. This self-fertilizing nature of legumes basically makes them valuable food plants in areas with very poor soil and in some countries with limited resources, as like Pakistan. It has been shown that crop health and yield can be improved by inoculating legume seeds with paver culture of symbiotic bacteria or rhizobia, because the soil is often deficient in the proper strains for forming suitable nodules. The second one in the nitrogen cycle is ammonification. In other parts, or simply we can say the ammonification, and in the nitrogen cycle, the nitrogen containing organic matter is decomposed by various bacterial species like Clostridium and Proteus, for example, that live in the soil and water. And as you know that the organic waste consists of large amount of protein and nucleic acids from dead bodies or dead organic biomasses, and as well as some nitrogenous animal waste such as urea and uric acid. The decomposition of these substances basically split off amino groups and produce ammonium, and this process of decomposition of amino acid, splitting of amino acid and the production of ammonium is termed as the ammonification process. The ammonium released can be reused by certain plants or converted to other nitrogenous compounds. The third one in the nitrogen cycle is the nitrification process. Simply, we can say the oxidation of ammonium to nitrite and nitrate is called nitrification. It is essential conversion process. The nitrification is an important conversion process for producing the form of nitrogen, that is nitrate, that is most useful to living things. Basically, the nitrification reactions occurs in two phases and involve lithotrophic bacteria that is present in soil and water. In the first phase, certain gram-negative genera such as Nitrosomonas and Nitrosococcus species, what actually they do, they oxidize the ammonium to nitrite. Is for what purpose? Is the means of generating energy. Then the nitrite, the produced nitrite, is rapidly acted upon by the second group of nitrifieds like Nitrobacter and Nitrococcus, which perform the final oxidation of nitrite to nitrate, or simply we can say NO2 to NO3. Oxidation means adding of oxygen to NO2, so the resultant is the NO3. Nitrate can be assimilated into the protoplasm by a variety of organisms, including plants, fungi, and bacteria, while a large number of bacteria also use it as a source of oxygen. The fourth one, so it's not the third one, it is basically the fourth one. The fourth one in the nitrogen cycle is the denitrification. The nitrogen cycle is complete when nitrogen compounds are turned to the reservoir back in the air by reaction series that convert nitrate through different intermediate steps to the atmospheric nitrogen, as here you can see. In this reaction, nitrate into the atmospheric nitrogen, the nitrate converted into the nitrites are converted into the nitrous oxide and into the atmospheric nitrogen. Basically, these reactions, this process are also carried out in different steps. The first step involves the reduction of nitrate to nitrite, and this is so common that hundreds of different bacterial species can do this reaction. Similarly, some other species, Bacillus, Pseudomonas, Parallel species, they also carried out this denitrification process. After the reduction of the nitrates to the nitrite is further reduced to the nitric oxide, that is NO or N2O, and after the conversion of the nitrous oxide, the nitrous oxide is further reduced to the atmospheric nitrogen. This denitrification process can be increased with some factors, like, for example, when there is excess soil moisture and oxygen-free condition. Basically, in excess moisture, most of the conditions are anaerobic conditions. When the condition is anaerobic, so the denitrified bacteria or denitrified microbial colonies are active. They convert the ammonium and the nitrate to the atmospheric nitrogen. Similarly, the denitrification and the ASR, we can say the accumulation of nitrates are also increased with the increase of temperature. So this was all about for the nitrogen cycle. In next class